The fourth type of macromolecule are the nucleic acids. And what do you know about them? Well, if you see this picture here, you should automatically say, hey, that double helix is DNA. So our nucleic acids are important because they make up the DNA. So DNA and RNA are the two classes. DNA stands for deoxyribose nucleic acid, and RNA is ribonucleic acid. They hold the blueprint for everything you need to create yourself, okay? RNA is useful in helping DNA become a protein and to do other functions in the body. The building block or monomers, like we were talking about, of a nucleic acid are nucleotides. They all have this general structure, a phosphate group, a 5-carbon sugar, and a nitrogenous base. This sugar can be ribose or deoxyribose. So if you think back to the carbohydrates lecture where we've seen the pentose sugar, this can be ribose in RNA or deoxyribose in DNA. And then this nitrogenous base can be adenine, guanine, cytosine, or thymine in DNA, or adenine, uracil, guanine, and cytosine in RNA. Okay, so ATGU DNA, AGUC RNA. Here's that 5-carbon sugar, and you'll see deoxy without oxygen is the difference between one. There's an OH group, here is just the H group. Okay, and these are monosaccharides, those one-ring sugars. Here's our nitrogenous bases. So they come in two types, purines and pyrimidines. So our purines are 2-ring structures, adenine and guanine. Our pyrimidines are 1-ring structures, uracil and RNA, thymine and DNA, and then cytosine in both. Okay. So when you join together these nucleic acids, you're going to have a phosphodiester bond from one nucleic acid to the other when you connect the sugar and the phosphate group, and that's going to form the backbone of DNA. In the middle, if we join 2 strands of nucleic acids together, we form hydrogen bonds. Okay, hydrogen bonds form between the nitrogenous bases. So phosphodiester on the backbone, nitrogenous base in the, hydrogen bond in the middle. So if we compare DNA to RNA, we know that the sugar is different. We have deoxyribose. The secondary structure tends to be a double helix, so it's usually 2 strands of DNA that run anti-parallel to each other. What does that mean? So if you take a chemistry class, you learn about numbering the carbons. One's going to have a 5' end up, and it connects to the 3'. So one runs 5 to 3, and the other runs 3 to 5, and that makes it anti-parallel. And then thymine binds to adenine, and guanine binds to cytosine. Between A and T, there are 2 bonds. Between C and G, there are 3 bonds. For RNA now, you have a ribose instead of deoxyribose for your pentose sugar. This usually exists as a single-stranded molecule, so you'll have those phosphodiester bonds, but you don't necessarily have the hydrogen bonds between 2 strands. And then instead of a T, you have a U here, okay? So one of the most important roles of RNA is to serve as messenger RNA. So you have your DNA, and it's composed of these genes, but how do you express those genes? That's where RNA comes into play. So you'll read that DNA molecule and create an RNA strand that's complementary to it. And so that means anywhere you see a C, you're putting a G, and T to A. Okay, so C and G binds together, A and T binds together, or A and U binds together when you're creating it as well. And I think that's it for the basics, and we will cover DNA further when we talk about genetics.